Assalamualaikum and very good day to everyone. My name is Dr. Wang. The topic that I'm going to present to you today is autosomal disorders. At the end of this lecture, you should be able to understand the meaning of autosomal inheritance, its inheritance patterns, which include autosomal dominant and autosomal recessive, the characteristics of each inheritance pattern, know how to perform genetic analysis by using Punnett square and pedigree chart, and some of the examples of autosomal disorders. First and foremost, let's recap some of the terminology. A gene is a sequence of nucleotides that codes for a specific protein. Chromosomes are made out of DNA and protein. Each chromosome contains many genes. Chromosomes come in pairs, one set from the mother, the other set from the father. Genes reside at a specific location on the chromosome, which is called locus. The alternate form of a gene at a given locus is an allele. Genotype is the genetic composition of an individual, whereas phenotype is the observable traits or characteristics of an individual. Homozygous is the two alleles being identical, whereas heterozygous is the two alleles having different nucleotide sequences, which may be caused by mutations. What is genetic disorder? Genetic disorder is an illness that's caused by one or more abnormalities in the genome. It is rare and can be passed down from one generation to the next. There are four types of genetic disorder, namely single gene disorder, mitochondrial disorder, chromosomal disorder, and multifactorial disorder. Single gene disorder is caused by mutations in one gene. Chromosomal abnormalities is caused by the entire or certain segment of the chromosome being missing, duplicated, or otherwise altered. Mitochondrial disorders are caused by mutation of DNA located in the mitochondria. Multifactorial disorders are due to mutations in multiple genes in combination with environmental factors. What is single gene disorder? Single gene disorder is caused by a single mutated gene. It follows the Mendelian trait, and the trait of characteristics can be passed from one generation to the next. Chromosomes exist in pairs, one from the father, another from the mother. Therefore, a gene for a particular characteristic is present twice in a cell, except for the sex chromosome in males. Human has 23 pairs of chromosomes. A mutated gene may be present either at the autosomes or at the sex chromosomes. An allele contains gene that codes for a characteristic. An allele can be either dominant or recessive. A dominant allele produces a phenotype when pair allele is in a homozygous or heterozygous genotype, whereas a recessive allele only produces phenotype when pair allele is in a homozygous genotype. Single gene disorder includes autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, Y-linked, codominance, and mitochondria disorder. An example for codominance is the ABO blood group. Here is an overview of the single gene disorders. Their characteristics and examples are listed here. Please go through. What is autosomal disorder? Autosomal disorder is a pattern of inheritance in which the transmission of traits, characteristics, or disorders depends on the presence or absence of certain alleles on the autosome. The transmission is not dependent on gender. Both male and females can express the disease. The inheritance pattern may be dominant or recessive. Here are the characteristics for autosomal dominant disorder. One mutated allele is sufficient for a person to be affected by the disorder. An affected person has an affected parent, which can be either the father or mother. An affected parent has a 50% chance of passing the mutated allele to their offspring. Both male and female are equally affected. It tends to occur in every generation of an affected family. A dominant allele is denoted by a capital letter, whereas a recessive allele is denoted by a small letter. Examples of autosomal dominant disorders are such as familial hypercholesterolemia, adult polycystic kidney disease. Both of these are common autosomal dominant disorders. Others such as Huntington disease, hypercalcemia, Marfan syndrome, achondroplasia, Ehlers-Danlos syndrome are rare disorders. Next is autosomal recessive disorders. Here are the characteristics for autosomal recessive disorders. A person requires 2 mutated copies of the gene to express the phenotype. An affected person usually has unaffected parents who are carriers, meaning each parent carries a single copy of the mutated gene. The expression of the disorder may skip generation, meaning it does not express in every generation. Both male and female are equally affected. Examples of autosomal recessive disorders are such as cystic fibrosis, phenylketonuria, thalassemia, albinism, sickle cell anemia. Others such as Bloom syndrome and Tay-Sachs disease are rare disorders. This diagram shows the comparison between autosomal dominant and autosomal recessive inheritance pattern in relation to gender and the mode of transmission. Genetic analysis allows analysis of genetic inheritance in human families. This is very useful, especially for married couple who has family history of certain genetic disorder. They might want to go for genetic counseling to determine the probability to have a healthy and unaffected baby. This can be done by simple genetic analysis such as Punnett square or pedigree chart analysis. A Punnett square shows the genotype of 2 individuals can produce when they crossed. A pedigree chart displays a family tree and shows the members of the family who are affected by the disorder. I will explain further in subsequent slides on how to perform the genetic analysis. This diagram shows a Punnett square analysis of autosomal dominant disorder for an affected father and a normal mother. Since it is an autosomal dominant disorder, we assign capital A as the abnormal or mutated allele and the small a as the normal allele. The genotype for the father is heterozygous. This is because it is very rare for a person to have both abnormal dominant alleles, as the chances for 2 persons with genetic disorders to get married are very slim. However, it is not wrong to assign the homozygous genotype, which is 2 capital A, for the father. Since the mother is normal, the genotype for an unaffected mother is 2 small a. When an affected father is married to a normal mother, the probability of their offspring to inherit the disorder is 0.5, which is 50%. This diagram shows a Punnett square analysis of autosomal recessive disorder for both parents who are carriers of a disorder. Since it is a recessive disorder, we assign the capital A as the normal allele and a small a as the abnormal or mutated allele. The genotype for both the father and the mother is heterozygous, which consists of one capital A and one small a. When a carrier father married to a carrier mother, the probability of the offspring to inherit the disorder is 0.25, which is 25%. Pedigree chart is another method for performing the genetic analysis. The diagram here shows the signs and symbols used in pedigree analysis. The most common symbols to use in a pedigree chart are such as the symbol for male, female, affected, carrier, mating, and siblings. Please remember that. In the pedigree chart, a numbering system is used to identify an individual. The Roman numerals indicating the generations. For example, the grandparent, the parents, and the children, whereas the Arabic numerals indicating an individual in a generation. Here is an example of a pedigree chart for an autosomal dominant disorder. How do we know it is an autosomal dominant disorder? The clue is the trait or characteristics can be seen in every generation. Whereas for an autosomal recessive disorder, the trait or characteristic skip generations, or it does not present in every generation. In this example, two children show the trait, but the trait was not present in the parents. Based on this information, we can conclude that it is an autosomal recessive disorder, and both parents are carriers. In this example, the trait occurs in every generation. Therefore, we can conclude that it is an autosomal dominant disorder. What is proband? The first member in the family history being diagnosed with a certain disorder and who seek medical attention is called proband. In the next few slides, I will present some examples of autosomal disorder. Huntington disease is an autosomal dominant disorder. This disorder affects the nervous system, which results in an eventual loss of both mental and physical control. It is rare and more common among white Europeans. The initial symptoms occur around 30 to 50 years old, and death comes on average 12 years after the onset of the disorder. Huntington disease is caused by mutation in the Huntington gene, in which the nucleotide of CAG, cytosine, adenine, guanine, repeats more than 40 times. This eventually leads to the production of an abnormally long of the Huntingtin protein. The elongated protein is cut or disintegrated into smaller toxic fragments that bind together and accumulate in neurons, disrupting the normal functions of the nerve cells. Huntingtin protein play an important role in the normal development of nerve cells in the brain. Marfan syndrome is another example of an autosomal dominant disorder. This disorder is caused by mutation of gene fibrillin 1. This gene plays an important role as the building block for elastic tissue in the body. Mutation of the gene will lead to too much growth of the long bones of the body. Sickle cell anemia is an example of autosomal recessive disorder. It is a disorder in which the red blood cells, normally disc-shaped, become crescent or sickle-shaped. As a result, they won't be able to carry oxygen throughout the body and have tendency to form small blood clots. These clots give rise to recurrent painful episodes called sickle cell pain cries. This disorder is caused by mutation of the hemoglobin gene, in which nucleotide adenine is changed to thymine. Carriers of this mutation has an advantage of protective against malaria. This is because the sickle cell infected with Plasmodium falciparum collapse and preventing the parasite from interfering with the host cell protein. This diagram here shows the inheritance pattern of sickle cell anemia. From here, we can see that a normal person have normal hemoglobin, whereas those with sickle cell anemia, the hemoglobin is appear to be clumped. Cystic fibrosis is a common autosomal recessive disorder. It is caused by the mutation of both alleles that code for the protein cystic fibrosis transmembrane conductance regulator, which is also called CFTR. This regulator plays an important role in the production of sweat, digestive fluids, and mucus. When CFTR is not functional, secretions, which are usually thin, instead become thick. This eventually leads to bacterial infection, inflammation of lungs, and elevated level of electrolytes in sweat. This condition can be diagnosed by a sweat test and genetic testing. Phenylketonuria, or PKU, is another common autosomal recessive disorder. PKU is a metabolic disorder in which an enzyme called phenylalanine hydroxylase, which is required to break down excessive phenylalanine from food, is deficient. It is caused by mutation of phenylalanine hydroxylase gene. As a result, excess phenylalanine from food accumulates in the blood, causing mosey body odor. In severe cases, accumulated phenylalanine will affect the brain function and development, causing mental retardation. People with PKU are advised to follow a low-protein diet. This is the end of my lecture. Let's go through some quiz. The alternate forms of a gene on homologous chromosomes are called allele. Which of the following correctly identifies the mode of inheritance for the given genetic disease? A. Cystic fibrosis recessive. Correct. B. Albinism recessive. Correct. C. Sickle cell anemia dominant. False. D. Adult polycystic kidney disease dominant. True. E. Huntington disease dominant. True. Identify a Mendelian disorder from the following: Down syndrome, phenylketonuria, sickle cell anemia follow the Mendelian disorder, whereas Klinefelter syndrome, 47XXY, Turner syndrome, 45XO, belong to numerical chromosomal abnormality. A couple, both carriers of albinism, have 4 children. One of their children is albino, and the remaining 3 are normally pigmented. What is the probability that their next child will be albino? Albinism is an autosomal recessive disorder. Based on the Punnett square here, we can see that the probability that their next child will be albino is 25%. Thank you and have a good day. Bye.